Hi, I'm Savannah. Thank you for allowing me to speak today. Today I'm going to be talking about the role of SlDML2 gene expression in ripening initiation of tomato fruit. So first, I want to give you a little bit of background about the importance of SlDML2 as a ripening control via its function in DNA demethylation. Next, I'm going to talk about the different components of my summer project, including some SlDML2 overexpression lines I worked with, bisulfite PCR analysis of those overexpression lines, and a CRISPR-Cas9 mediated SlDML2 knockout that I created. So why would we study the tomato ripening process? We know that today, post-harvest fruit loss is one of the most limiting factors on our food production and distribution systems. According to the FAO, in fact, about 50% of fruit is lost worldwide due to these post-harvest processes. This is an extreme problem, especially when we take into consideration the fact that we'll have approximately 9.6 billion people on the planet in the year 2050. And we believe that being able to control the ripening process could alleviate some of this fruit loss. So we're looking at tomato specifically because tomato fruit ripening research can be applied to a lot of different species of fleshy fruits. So this is just an overview of the tomato fruit development and ripening process. We start with anthesis here, which is when the flower opens. The fruit grows until it reaches mature green, where seeds are mature. Then we have breaker, and then we eventually have breaker plus 7, which is 7 days after breaker. And we consider this red ripe. Below here we have our tissue-specific ripening pattern. So ripening begins in the locule, which is this gel-like tissue here, and then it moves into the rest of the fruit, including the pericarp. So what are some of the factors that affect fruit ripening in tomato? First, we have transcriptional regulation. So these are all mutants where a transcriptional regulatory factor has been disrupted in some way, and as you can see, they do not undergo proper ripening. We also have ethylene signaling inhibition. This is a never ripe mutant, which is partially ethylene insensitive, so it also does not undergo proper ripening. And finally, we have this SlDML2 mutant, in which SlDML2 is not being properly expressed, and it does not undergo DNA demethylation. So out of these three, DNA demethylation is the least well-known and researched, and that is what I have been focusing on in my research project. So we know that genome-wide hypomethylation occurs during ripening. Here I have a chart from a previous study that shows the differences in methylation between the different stages of fruit development. You can see there's a very big difference between the methylation level of fruit at 17 days post-anthesis as compared to fruit at red ripe, which is here. And we also know that this demethylation occurs at discrete loci, and these loci tend to contain ripening-related transcription factors, binding sites for these ripening-related transcription factors. So here this shows one such place, the CNR promoter. As these peaks grow smaller, that represents demethylation in this DMR region. And we also can show that CNR and RIN mutants don't exhibit the same loss of methylation in these regions because they are not affected by DNA demethylation. Okay, so SlDML2 encodes a Demeter-like DNA demethylase. We know that SlDML2 is highly expressed during the fruit ripening process. Here you can see that at 20 days post-anthesis, there's not a super high expression of SlDML2, but its expression exponentially increases during the process. And then again, I'd like to point out that SlDML2 mutants, which this is a mutant in a microtome background, does not undergo ripening. And here we have a wild type fruit for comparison, and there's a very big difference here. So for the first part of my project, I was examining SlDML2 overexpression lines done with a 35S promoter. And we previously, before I arrived this summer, 2 overexpression lines have successfully been created in my lab by my mentor, Dan Evanich. And those are overexpression 13 and overexpression 5. We know that these lines are successfully overexpressing SlDML2 because of analysis of tissue at different stages and in different tissues during the ripening process. Here we have a graph that shows that SlDML2 is more highly expressed in both the overexpression 5 and the overexpression 13 lines than the wild type fruit at mature green. I would also like to point out that overexpression 13 is more highly expressed than either the overexpression 5 or the wild type. Despite the success of generating these overexpression lines, we don't really see a big difference in the phenotype of the fruit. We have an SlDML2 azygous fruit on the left, which has segregated out the transgene, so it is not showing any phenotype of overexpression of SlDML2, whereas we have the overexpression 5 and overexpression 13 lines, and they look pretty much the same as the azygous fruit. So not a lot of obvious phenotypic difference there. However, despite this lack of phenotypic difference between our fruit, we wanted to investigate further what was occurring on the molecular level. And in order to do this, I conducted bisulfite PCR analysis of our SlDML2 overexpression lines. We chose to convert the NOR promoter fragment in these overexpression lines due to the fact that we know that it is demethylated during the fruit ripening process, which is shown here in this chart. These peaks, again, represent the levels of methylation, and they quite clearly decrease during the ripening process. For those of you who don't know, bisulfite conversion of DNA works by converting unmethylated cytosine into uracil, which is then converted into thymine during the PCR amplification process. So these are my bisulfite PCR results for the percent methylation of cytosine at the mature green stage in pericarp tissue of our 2 overexpression lines and the wild type. So our wild type is in pink, our overexpression 5 is in green, and overexpression 13 is in gray. I'd like to point out that overexpression 13 is definitely lower, has lower methylation levels than either overexpression 5 or the wild type. And this sort of correlates with our previous data that shows that overexpression 13 is probably more highly expressing SlDML2 than the other 2. So that makes sense. But overall, we were able to show a difference in methylation due to the overexpression of SlDML2. So in addition to our overexpression lines, I created a CRISPR-Cas9 SlDML2 knockout. This is just an image of the construct that I was making. We have with our guide RNA here, which targets the SlDML2 gene. And the reason I created this construct was because our lab does not actually have an SlDML2 CRISPR knockout in an AC background, which is the wild type that we use in our lab. So having this mutant would be very applicable to all of the research done in our lab. And it will also allow us to further investigate the function of the gene by seeing what the silencing of this gene, how that affects phenotype of fruit and methylation levels. And we expect that the knockout will not undergo ripening at all. This is just a small diagram to show that the guide RNA will target our SlDML2 gene and cause a double-stranded break in the gene sequence. So I did successfully create this construct and cloned it as well. Here I've got the guide RNA that I used, that I inserted into the construct, compared with a Sanger sequencing result from my cloned colonies. And as you can see, the guide RNA is completely there, so I was very successful in this. And this is just, this just shows that SlDML2 falls at the beginning of this gene sequence, so it's going to have the best chance of disrupting the domains involved in DNA demethylation. So in summary, we've shown the importance of SlDML2 as a ripening control via DNA demethylation. We've shown that SlDML2 expression lines do have differences in methylation levels via bisulfite PCR, and I have successfully created my CRISPR knockout construct. So in the future, my knockout construct will be sent for transformation, and it will be used in further research for its effect on ripening. My mentor has also created fruit-specific constructs, which will be sent for transformation since we have reason to believe that overexpression of SlDML2 may be lethal. So this will only overexpress SlDML2 in the fruit. Further bisulfite PCR analyses will be done for multiple replicates and different tissue at different stages of our lines, as well as future crosses. Well, we've been working on crosses between our overexpression lines and ripening mutants to see if we can rescue the phenotype. And I've been working on that as well, so if you have questions about that, please feel free to ask me later. And I would like to thank Dr. James Giovannoni for allowing me to work in his lab, my wonderful mentor, Daniel Evanich, as well as everyone else in the Giovannoni lab for being a great help and support, the BTAT greenhouse staff, and Delaney and George Jander for making this possible. Does anyone have any questions?
No questions about tomato ripening?
Yes.
How many replicates can you do for the bisulfite PCR?
I just did the one so far because we've just harvested our fruit. But I've collected a lot of tissue for multiple replicates of the pericarp and the locule of both mature green, breaker +3, and breaker +7 for all three of the lines. I've done bisulfite conversions on multiple replicates for the MG pericarp, but I was only able to do the full analysis on one replicate of that. I think overall the goal would be to do both in order to, I think depressing the ripening process would be more important in preventing the spoilage of fruit during food distribution systems and that sort of thing. But overall, for my specific project, we just want to understand the role of this gene in order to be able to use that for further applications in the future.
Okay, well, thank you.